We will wait for 2 more minutes. At 6:05 we will start. We can start now. So I welcome you all in this live session. It is a week one assignment live session while I will be solving the week one assignments, but the questions here will be different from the assignments that you are solving this time. That the questions that were given in the week one assignment this time, the questions are different. So my name is Ubaid Tariq, and I'm a PMRF scholar at BSB Department of IIT Kanpur. And in this in this cell culture technologies problem solving session, I will be conducting this live session every Tuesday at 6 p.m. So you all are welcome to join these live sessions. Some more people are joining, but we will start the presentation. So the first question is, which of the following is not the phase of cell cycle? You can also make this session interactive, and you can also give the answers in the chat box. This is a very easy question. Which of the following is not the phase of cell cycle? The answers are interphase, mitosis, gap zero, open phase. Are you able to see the presentation? Because someone was saying I'm not able to see. Okay. So the answer is 4, open phase. Open phase is the wrong answer. So we'll explain you how. I will explain you about the cell cycle here. A lot of people are joining. I will explain you cell cycle. The cell cycle has majorly two phases. One is the interphase, one is the metaphase. Interphase is the phase here. I don't know if there is any pointer in this Google Meet. So I think you are able to see the cursor. So interphase is the phase, is the preparatory phase, and this mitotic phase, which consists of these five phases, they consist of the mitotic phase. So interphase, in this the cell prepares for it for the division, and here in these phases cell actually divides. So interphase is a preparation phase, prophase, in which this spindle formation starts in this prophase, and spindles also start to move at the two junctions or two ends of the cell. In the metaphase, this metaphase plate is formed. You can see here the chromosomes, they are aligned in a one line called the metaphasic plate. And also the spindle fibers and centromeres, they join to the centromere of these chromosomes, and this metaphase plate gets formed. And after that there is another phase called anaphase. To anaphase, these sister chromatids, in case of mitosis here, I'm explaining the cells like mitosis. So these sister chromatids, they are separated from each other, like these chromatid fibers, they shrink, and they will pull these sister chromatids apart. So another phase after this is a telophase. In the telophase, these chromatids, when they get separated in the anaphase, the this nuclear membrane will be formed, and these chromatids will get incorporated in the nuclear membrane, and after this cytokinesis occurs. So cytokinesis is the formation of division of the cytoplasm. So here only the open phase is it's not the any phase of cell cycle. The interphase, you are also seeing the G zero phase, gap phase, zero phase. This G phase is also the part of the interphase, and mitosis is mitosis, and interphase I already explained. Sorry. Second question is DNA replication occurs under which phase of cell cycle? DNA replication occurs in which phase of cell cycle? Can anyone answer? Yes, the answer is 3, synthesis or S phase. So in the earlier slide, I showed you details of this mitosis phase, like the interphase, the prophase, anaphase, telophase, cytokinesis. That all they come under this mitosis or mitotic phase in which they divide. But I didn't explain the interphase. So here in this slide, I have given the details of interphase. So in the interphase, there is the first phase called G1 phase or first growth phase. In this G1 phase, cell decides that it should go for division or it should not go for division, and this cell has already completed the one cycle of this division, and after cytokinesis the next G1 phase starts. So if the cell needs to divide, the cell prepares itself for the division, and cell also prepares itself for the synthesis of the DNA or duplication of the DNA. So if the cell is not ready for the division, or cell wants to go in a dormant phase, those cells which do not divide, so the cell will go into the G0 phase. But if the cell decides to go for the cell cycle, then the cell goes in the synthesis phase. So here in the synthesis phase, which is the second phase of this, the DNA synthesis occurs. So here the number of chromosomes does not double. I don't know, is there any pen option or so that I will be able to show. So I will explain like this. So in the synthesis phase, the number of chromosomes does not double. So the n is 46, as we have 46 number of chromosomes. But the C, the total number of genes, they are doubled. For example, here you can see that earlier in the G1 phase and earlier in the G1 phase, this is the chromosome, and as we are diploid, so we have two sets of one chromosome. So this chromosome is intact, but at the time of synthesis, this phase, at the time of synthesis, what happens? The DNA in the chromosome, it doubles, and here you can see two sister chromatids in a one chromosome. So we have two sets of chromosome. For example, this green one has a two. We have two sets of this green one. So here the n will be 46 because we have two sets. 2n will be 23, but the DNA content will double. But both the sister chromatids will remain adherent to each other in the synthesis phase. After this phase, the G2 phase occurs, and in the G2 phase, the cell also checks if the synthesis has occurred properly or not. So the cell checks if for the mutations, if there are any mutations, or if the synthesis occurred or not. So after that, if the cell decides to divide, then the cell goes in the mitosis phase, and in the mitosis phase, ultimately the cell divides. Next question is on the differentiation, potency of cells dash increase, decrease, do not change, none of the above. Anyone knows the answer?
Do not change.
Does not change. Okay, anyone else? On it's not the differentiation, it is differentiation. So most of are you saying increase? Some also said decrease, but the answer is increase. So potency is the ability of the cell to form the different types of cells in a differentiated form. You may have heard about the multipotent, pluripotent, totipotent. Totipotent means the cell can divide in any type of cell, which may be the germ cell or any type of somatic cell. Pluripotent means cell can divide into any type of cell, but only germ cells, like three germ layers. My cell can divide into three germ layers: ectoderm, mesoderm, and endoderm. And multipotent means cells can divide into some few number of cells, or for example 2 or 4 cells. And unipotent means cell can only divide into one type of cell. For example, cardiac progenitor cells or cardiac stem cells. Cardiac stem cells are unipotent. They only divide into cardiomyocytes or cardiac cells. But totipotent cell or pluripotent cell can divide into any type of cell. So someone is saying repeat. So I have to repeat the potency? Yes. So potency is the ability of the cell. For example, when the cell, you know that certain stem cells have the property that they can grow into different number of cells after differentiation. So ability of the cell to grow into different diversity of the cells is called the potency. If the cell has a more ability to divide or differentiate into large number of cells, large different types of cells, then they are called, their potency is more. If some cell, like I have given the example of cardiac stem cells. Cardiac stem cells can differentiate into only one type of cell, that is cardiomyocyte. So its potency is very less. But the totipotent cells can divide into any type of cells. So it has the highest potency. So it can totipotent cell, if the stimulus is given to the totipotent cell, so it can make any type of cell. So it has the highest potency. After that, second in the second number is pluripotent, which can also make the large number of cells, like all the germ cells, all the three germ layers, ectoderm, mesoderm, endoderm, but it cannot make the non-germ layers. So here the pluripotent has the less potency as compared to totipotent. And also the multipotent can also multipotent cells can also make fewer number of cells, or they can differentiate into fewer number of cells. They have the less potency as compared to pluripotent. So potency is the ability of the cell to differentiate into different number of cells, different types of cells, sorry. So here I will explain the dedifferentiation and redifferentiation. So dedifferentiation is a process in which the normal cell, which is the somatic cell, like the cell of our any type of body, for example fibroblast, cardiac cell, hepatocyte, any type of cell, they don't our the somatic cells present in our body, they don't have the property of potency. They do not, they will not be differentiated into any other type of cell. They only divide. But our fibroblast cannot make the cardiomyocyte, or our hepatocyte cannot make the epithelial cell. So dedifferentiation is a process in which these somatic cells can be dedifferentiated into the cell or stem cell which has a more potency, like induced pluripotent stem cells. Induced pluripotent stem cells are the cells in which using the process of dedifferentiation, they are dedifferentiated from fibroblasts to pluripotent cells, and now these pluripotent cells can make any type of cell. Here this is the somatic cell, and after dedifferentiation the stem cell like state or transient state is made, and now if the stimulus is given, this cell can make any type of cell in the cell cycle, or in some cases this cell also may die, or this cell may also redifferentiate. Redifferentiate means it can also differentiate and make the same somatic cell. So it is the opposite of dedifferentiation. Any question regarding this? Any doubt? Repeat the answer of first question. The answer of first question is open phase. Which one of the following is not the phase of cell cycle? So open phase is the answer. I have explained all the phases of cell cycle. So open phase is not any phase. If sometimes I miss the chat box, you can directly unmute yourself and you can ask. So fourth question on the differentiation, it is question related to the same topic that I was keep explaining. On the differentiation, function of cell becomes less specialized, more specialized, unknown, has no correlation with the specialization. Yes, more specialization. As no, no. The answer is less specialized. On the differentiation, after the differentiation.
Not redifferentiation.
So after the differentiation, what happens? For example, fibroblast or cardiomyocyte, it has a more specialized function. It functions only, uh, what I can say, it only contract. So it,
So,
yes.
but the question says differentiation, not
Oh,
dedifferentiation.
sorry. The question is differentiation. So yes. On differentiation, function of cell becomes more specialized. Yes, because stem cell has a more stem cell can make any type of cell. So the answer is more specialized. For example, if the stem cell makes the fibroblast, then the function of fibroblast is more specialized. as compared to the stem cell. But the question can also be like this: on dedifferentiation, function of cell can become what will be the answer? Less specialized, because in the dedifferentiation, uh, differentiated cell is converted into stem cell like behavior. So in that case, less specialized behavior. Sorry, I misplayed it. So question number 5. Which of the following is a marker of induced pluripotency?
All of them, right?
All of them, no. Actually, I have mentioned here also, you can see.
Third one.
Yes, third one, OCT4. This this slide that I am showing you here, it was a breakthrough in the stem cell therapy or stem cells. Earlier, when we need to when we need stem cells in for any type of research or for any type of therapy in the clinics, always we before 2006 always we needed to isolate the stem cells because stem cells are very less. They are maybe present in our this in our bone marrows, and they also are present in some tissues, but the number is very less. Only in our bone marrow there is a more number of bone derived mesenchymal stem cells. So earlier it was very difficult to isolate the stem cells, and there was no dedifferentiation method was not discovered that time before 2006. And the group from Japan, they discovered the dedifferentiation potential, and they made these induced pluripotent stem cells. These so they made induced pluripotent stem cells. So what they did, they took fibroblasts and induced or injected through the lentiviruses these four transcription factors. These transcription factors, they elicted some of the pathways in the cell, and when the cell were grown and in and after that the differentiated or induced pluripotent cells were isolated. So now we don't need to isolate pluripotent cells directly from the person or animal or whatever be the source. So we can also de-differentiate the fibroblasts or so any type of somatic cell so that the pluripotent cell is made, and from this pluripotent cells we can make any type of cell. For example, this is the fibroblast. Earlier we were not able to make from the fibroblasts. We were not earlier it was not possible from the fibroblast to make the nerve cell, or from the fibroblast to make the blood cell or the cardiac cell. Now through this induced pluripotency, from fibroblast or cardiac cell or any other cell, you can make any type of cell through this through these transcription factors. So after this dedifferentiation here, dedifferentiation step through these transcription factors, pluripotent stem cells are made, and in the next step, the differentiating some differentiating markers are given. For example, specialized differentiating markers for the neural cells. There are some specific differentiating markers for the cardiac cell, blood cells, and treatment of these markers will be given, and we can get any type of cell.
Excuse me, sir. Will this
Yes.
session help us to attend the assignment questions from week one?
What was your doubt? Can you repeat?
This session is helpful in attending the questions from week one assignment, sir.
Yes, but week one assignment, we're advised to give some more questions besides those questions. So those questions you have to solve by yourself. So
Sir, that only, sir. Where are the lecture materials are available for
what can you repeat? Sorry.
the, where we can get the lecture materials, sir?
So you have already lecture material is up. Videos are already uploaded in the NPTEL.
Videos are there, sir, but No, notes materials are not uploaded, sir.
Okay, maybe
Like
after some
other
time,
subjects.
maybe after some time they will upload. You can raise a query in that Google. There is some there is some which discussion form is there. You can raise your query and maybe they will answer.
But tomorrow is last date for submission of week one assignments, sir.
Okay. So if you have any doubt regarding that also, you can also ask here after I will finish these questions. Eh?
Okay, sir. Thank you.
Mhm. So GNDP is a growth factor that stands for. What is the answer? Anyone? Yes, 2 is the answer. Glial cell line derived neurotrophic factor. This is the neurotrophic factor that is very important for the growth of neurons. Without these neurotrophic factor, these neurons will not grow properly. So this neurotrophic factor or growth factor is important when we grow these nerve cells in a cell tissue culture. So we must have to add this neurotrophic factor so that these nerve cells will grow in the cell culture. Question number 6. Which of the following proteins is responsible for the force generation in skeletal muscle?
Myosin.
Yes, anyone? Myosin, yes. Can you name any other protein which has a main role? Yes, actin. Actin and myosin are two important proteins that have the role. Like here in this picture, this actin is called the thick filament, and this myosin is a thin filament. So they are attached to each other. Actually, on this actin there is a receptor for the myosin, and they are attached to each other. Earlier, before contraction, they are not attached, but in the presence of calcium, which is important for the contraction, and other proteins, they get attached. And when the signal is received, after that they slide with each other, like here. You can see they slide with each other, and which will lead to the muscle contraction. Uh, Actually, I don't have the pointer from the next session. I will keep the look in that. So here, these are called Z discs. These here, one is a Z disc, one this section, and in each Z disc there are these are called sarco. I think sarcomeres. So they are present in this Z disc and they help in the contraction. So the contraction of they are present parallelly in the muscle. So when they contract, so these muscles, let it be cardiomyocytes or skeletal muscles, they only contract in one axis. For example, if you are seeing my hand and this is the length of the cardiomyocyte or skeletal muscle, they are present here and it can. For example, this is the cardio muscle and it can contract like this axis only, yeah? Because of the contraction of these actin and myosin, because of the sliding of these actin. Another question is actin is a sugar, lipid, carbohydrate or protein? Yes, the answer is protein. Actin is a protein which, as I already said, which has a role in muscle contraction. So this actin myosin they are present in all the cells which have the contractile property. Can anyone tell me in our body how many types of cells have contract? general cells, name of the cells. You can unmute yourself, anyone, or you can write in the chat box. Am I audible? Yes, cardiomyocytes is one type of cell that contract another. So these actin myosin, Nani, I am talking about human. Give me the cells present in the human body, different types of cells that have the contractile ability. And in all these types of cells, there is this these are discs are present. Yes, skeletal muscle cells. And third type, there are three types. Maybe there are some other. You are missing the one important cell. Skeletal muscle, cardiomyocyte, and third is a smooth muscle cell. Yes, smooth muscle cells. Smooth muscle cells are present in our blood vessels, in our GI tract, and in our GI tract and blood vessels they also contract. So there in our GI tract or blood vessels, these smooth muscles are present that will help in the contraction. And all these, uh, whether it be smooth muscle cells or cardiomyocytes or the skeletal muscle cells, this system is present for the contraction. So they have the same system. So another question is, in heart, cardiac cells contract in a unison by dash through a gap junction. They contract in a synchronous manner at a one time. So there are four answers given: cell wall current transfer, cell membrane current blockade, cell to cell current transfer, cell wall current blockade. So the answer is three: cell to cell current transfer. As you can see in this picture, I have given that this is a picture of cardiomyocytes. So these cardiomyocytes, they are connected to each other. All the cardiomyocytes, they are connected to each other through these intercalated discs. And between these intercalated discs, there are gap junctions and there are also desmosomes. So through these gap junctions, these electrical impulses, they transmit from one cell to another cell. As you know that in case of heart, the synchronous contraction is very important. It is very important. For example, when the chamber of heart it contracts, the whole the number of cells, they actually contract in a milliseconds, like 200 to 0.1 second, or and this is the actual contraction time. So all the cells should contract at the same time. So in that case, these cells should be well connected to each other. So when the electrical signal is received, because the electrical signal which the cardiomyocytes receive, they receive from the conduction system like the SA node, AV node, Purkinje's fibers. They give the through these conduction system, they receive the electrical signal. But electrical signal does not go to every cell of the cardiomyocyte. There are billions of cardiomyocytes in the this heart. So these conduction system, these conduction wires, they do not go to each cardiomyocyte for the conduction at the same time, but they will go only at the few places in a whole heart. So after receiving the electrical signal, they have so much connectivity that this electrical signal, if it's received at here at this point, it is abruptly in a no time, you can say in a no time, it's transmitted to all the cardiomyocytes, and all the cardiomyocytes contract in a synchronous manner at the same time. And this is the main role of gap junctions in the cardiomyocytes, and through these gap junctions, cell to cell current transfer occurs very quickly. 10th question is, what are cardiac cells called? What is the name of cardiac cells? Yes, the answer is 4, cardiomyocytes. Cardiac cells actually in this in these answers, the answer is cardiomyocytes, but the cardiac cells are also made up of overall cardiac heart has many type of cells. They can also be called as cardiac cells. So here the answer is cardiac cells. So these cardiac cells, which are called cardiomyocytes, they actually have the contractile power, and mostly these cardiomyocytes are present in this myocardium, which is the middle layer of the heart. And because of these cardiomyocytes, the heart pumps and contracts. But there are also other types of cells in in this heart. For example, heart has three layers. Outer layer is the epicardium, middle layer is the myocardium, and inner layer is the endocardium. In the outer layer, there is also the pericardial layer, which is outside this layer. Pericardial pouch, we have heard about that. So heart is in the pericardial pouch, and between this epicardium and that pericardial pouch, there is the cavity called pericardial cavity, which contains the pericardial fluid. So you may have heard that these epithelial cells, they line the cavities of all our body, and also the cavities of organs, internal organs, surface of the internal organs. So here also the heart is covered by the epithelial cells, endothelial cells. So here here this is covered by endothelial cells, and also the fibrous tissue. It also contains this epicardium also contains these epithelial cells, which are called mesothelium, and they also are made up of some fibroblasts, fibrous tissue, which gives the strength to this epicardium. And the part which is contracting, epicardium is not contracting, and inside the endocardium is not contracting. Only the contractile power has the these myocardium has the contractile power, and it contains these cardiomyocytes. But you you may be surprised that these there are two major types of cells in this myocardium. One are cardiomyocytes, another are fibroblasts. Can anyone tell me the number of cardiomyocytes should be more, or number of fibroblasts should be more in the myocardium? It is a contractile ability. It is contracting because of cardiomyocytes only. So cardiomyocytes should be more, or fibroblasts? Why fibroblasts should be more? As it has a function of contraction, and only cardiomyocytes contract, fibroblasts do not contract. Why there should be more fibroblasts? Yes, is anyone answering? Actually, whoever was answering, he is right that there are more fibroblasts as compared to cardiomyocytes. So in the atrium there are 33% of cardiomyocytes, but here there are like 50-50 also. So there are fibroblasts are also present, because these cardiomyocytes, they only have the contractile power. Other types of things like secretion of extracellular matrix, and other signaling things. These fibroblasts do these signaling things, and without these signaling, in different disease conditions, in normal conditions, all these signaling is controlled by fibroblasts. And different, for example, signaling molecules, the fibroblasts secrete, they also affect the cardiomyocytes. So that's why they are also important in case of in this cardiac muscle, in case of myocardium, even though they are not contracting, they do not have the contractile power, but they are also present neck to neck. Their number is comparable to cardiomyocytes because they are important for signaling in the myocardium. And the inner layer is the endocardium, which is made up of epithelial cells. As you know that our every cavity of the body is made of is lined by epithelial cells. They have the endothelium, and it also has a fibrous layer of which gives the support to epithelium. There are also some important special type of cells which have the conductile abilities and which also have the pacemaker abilities. This pacemaker cell in the SA node is a very rare cell, and no other cell in the body has this type of function. It has the autonomous function of generating these electrical impulses without the influence of the brain. It generates the electrical impulse. So our heart is autonomous. If there are no nerve interventions, so heart can function. Heart can pump itself only. So these pacemaker cells, they are specialized cardiomyocytes only, and they have the autonomous generation of electrical impulse, and this electrical impulse is then moved through these different special types of cells. These special types of cells are also the cardiomyocytes, and they are special in the way that they do not have the contractile power. They are cardiomyocytes, they do not have the contractile power, and that's the reason is they do not have the sarcomere, like they do not have actin myosin. The expression is more, but the expression of different channels like ion channels, they are more in this type of cardiomyocytes, and that's why they are specialized for conduction. So this was all about these questions. If you have any doubt or you have any questions, you can ask
Sir,
here in this session. If I don't have answer, so I can answer you later. But I will try my best to answer.
sir,
Hmm. Yes.
sir, is there any chance to get any extra marks for answering in this chat?
Is there any you are getting extra marks? So here,
Sir. So
huh?
many students are answering in there, and I was silent for.
No,
So
no,
I
no, no,
asked.
no, no. There are not any the extra marks for
Okay,
answering
sir.
here.
Sir, also we just need to complete our assignments and tasks to give us.
What? So you have the assignment. Uh,
We
you
need to
can.
complete the assignments and any other tasks to get the marks only.
So you only have to give your assignments and attend this session. So here the answer, I do not count the marks for answering here. Actually, I can directly give the answer, but to make the session interactive, so I ask the answers from you also, you people also.
Okay,
But
sir.
if
No, I was just silent, and so many students are answering, so
oh,
I just asked.
no, it is okay. You can, but if you know something,
Okay, okay.
you can
Thank you,
write in
sir.
the chat box and you can answer.
Thank you, sir.
Hmm. So someone has given the question. Wait, I will. Wait, what was the question? I'm not able to see the chat box. In the cell culture, the extracellular matrix coating on the substrate is required for. Can you explain this question? Yes. As you know that the what is cell culture? Cell culture is a in vitro condition where we grow the cells that actually grow in our body. So the cells that we are growing in the cell culture, they actually grow in our body. They have they are adjusted, or they are they from they are in such a way they only grow in the encourage dependent manner. For example, most of the cells of our body, like whatever organ you name, the cells in that, they grow in an encourage dependent manner. But some type of cells, like blood cells, they go in an encourage independent manner, like in the cells in our blood, they don't need in our body also, they don't need to anchor, they are flowing. So in the cell culture they also grow in the same way. But the other cells of our body, they grow in an encourage dependent manner. Cell to cell connection and cell to cell, they are important for the growth of cells. And also they do not grow floating. They in our body, they are in the, or they are in an organ, and they are in contact with each other, and they are in contact with some support, like connective tissue and extracellular matrix. So extracellular matrix is important for the growth of cells, because every cell in our body, uh, they are attached to each other because of the extracellular matrix which is present. So if you take any organ or any tissue of our body, the extracellular means which is outside the cell, and this extracellular matrix is secreted by the cells only, and because of this extracellular matrix, it is giving the good environment for the cells to grow. That is why, if we want to grow the cells in a cell culture, so extracellular coating is important for the cells to grow. If the cell culture plates are not coated, the cells will not adhere, because they are anchorage dependent. They need to adhere to uh, the plate for the growth. But as in our body, they are adhered or connected to the extracellular matrix. So cell culture plate also, they will adhere or grow in the cell ECM, this coated matrix also. And nowadays there are some different proteins also that are coated in cell culture plates. But ECM or collagen is the major protein which is coated in the cell culture plates. Any other question? Any other question? If there is no question, then I can stop. Which is the usual marker of pH? In you're talking in cell culture. So in cell culture, for the pH, the phenol red is added in the cell culture media. That will help to detect the change or pH in the pH. So if pH is more acidic, this phenol red will change into a yellow color, and if it is more basic, it will change into the pinkish color. So we will able to by visualization, we will be able to know the change in the pH of in the cell culture. Any other question? Bromothymol. But it is used for I don't know about it. Bromo. I don't have any information about this. You can write the full question, so that if I don't know, I don't even understand the question. So you can write the full question, so that if I will not be able to answer this time, I can answer in the next session. So if there are no more questions, I will stop this session, and we will meet next week, next coming Tuesday at 6 p.m. Nee, but I don't exactly know about this, but most of the cell culture medias they contain this phenol red. They contain phenol red as an indicator for the pH. And as phenol red has an advantage that it can change color both in case of if the pH is changed to acidic or basic. So bromophenol it changes the color. So if it is if the color if the pH is acidic, so it changes to this yellowish, and if it is basic, it can change to this pinkish color. So thank you all for joining this session. I will see you next week. So these recordings they will be uploaded on YouTube. So you can note. I will upload them in the YouTube. So you can note my YouTube ID Ubaid Tariq. No, Ubaid But sorry. So here I will share you the link. I will share the link of my YouTube channel in the chat box. There are also some other lectures. You can also take a look of those lectures also. I don't know how to share. I don't know how to share this. Well, I will copy this URL, and you can also see this picture of mine. So I will paste here in the chat box. You can click on the link, and you can directly search Ubaid But. You can see there may be more persons in this name. So you can watch those videos, and there are some other videos also which may be helpful for you, so you can watch those also. So thank you for joining. We'll meet you in the next session. Thank you for joining and giving your time. Thank you. I will end the meeting now.